Gram staining is a differential staining procedure that turns gram-positive bacteria purple and gram-negative bacteria pink when visualized under a microscope. The stain can also help to determine the shape and morphology of the bacteria in a sample. To prepare a slide, a sterile inoculation loop is used to transfer bacteria to the slide. Bacteria from either an agar plate or a broth tube can be used. If bacteria from an agar plate is used, however, add 1 or 2 drops of distilled water on the slide before adding the bacteria from the agar plate. No additional water is required if the bacteria is coming from a broth tube. Allow the slide to completely air dry. To speed up this process, keep the slide near a Bunsen burner. Once the slide is air dried, heat fix the bacteria to the slide by passing the slide over the flame of the Bunsen burner 4 to 5 times. Now the slide is ready for staining. Before any stains are added, both gram-positive and gram-negative bacteria are colorless. First, the slide is flooded with crystal violet for 60 seconds. This stain passes through the cell walls of both gram-positive and gram-negative bacteria. This stains both types of bacteria purple. Next, the slide is flooded with iodine for 60 seconds. This forms a complex that essentially locks the crystal violet in place. Both types of bacteria are still purple. The decolorizer is applied to the slide for about 10 seconds. It degrades the outer membrane of gram-negative bacteria, which frees the crystal violet iodine complex only in gram-negative bacteria. This leaves the gram-positive bacteria purple and the gram-negative bacteria colorless. Finally, the slide is flooded with safranin for 60 seconds. This pink dye is absorbed by both gram-positive and gram-negative bacteria. However, the darker purple stain of gram-positive bacteria masks the lighter pink color. So under the microscope, gram-positive will be purple and gram-negative will be pink.